Okay, 1b on page 48. So what we're looking at with these type of questions, again, are polygenic questions, or polygenic questions. And these type of questions are different than what we've experienced before. We've always talked about, with dihybrids, we always talked about law of independent assortment. We said an allele for one particular trait separates independently of the other trait when they isolate themselves into gametes. But with polygenetic, we actually are getting some interaction happening between the two different genes located on different chromosomes. So this is 1b, of course. We've done 1a already. So let's kind of do a legend that we used in 1a. We said that if I have little c's in a recessive allele, this indicates that there's going to be no pigment present. So that means I don't care what these other alleles are going to be. These are going to interact. But when I have two little c's, this is going to produce an albino. No color whatsoever. So it's this here that's determining whether or not there's pigment, or in this case, no pigment is going to be produced. So I put these other lines in here indicating that I don't care about the other gene located on a different chromosome. I don't care if it's allele capital B for dominant or recessive little b. As long as I have little c's, it's going to produce no pigment, and that's going to give me an albino. So if I need pigment, or if I'm going to get pigment present, I must have the presence of one dominant allele for pigment, in this case a capital C. I don't care what the other allele is. As long as I have a big C, I'm going to have pigment being produced. Now, what color the pigment's going to be depends on this other gene located on a separate chromosome. And if that gene is represented with a dominant B, I don't care what the other allele is, that's going to give me black. Okay, so those mice are going to be have a black coat color. If I have a big C, doesn't matter what the other allele is, and I have two recessive little b's, that's going to be a tan or a brown mouse. And that's all we need for our legend for this. So again, polygenic, because you have one trait represented by a gene on one chromosome, will affect the expression of the other one. So these two little c's, that particular gene on a chromosome, will affect the other gene for color, whether it's black or tan. Okay, so when we see this type of question, all we're going to do is we're going to isolate that. We know the genotype of the parents. They actually want us to tell us the phenotypes of these two parents as well. So again, if we see two little c's, look in their legend, we know this is going to be an albino parent. And the other one has a capital C, so that means it's going to have color. What color depends on the capital B here, so that is going to be a black. Okay, so that's the actual phenotypes, which was partly what the question was asking. And then they want you to do the actual cross. So again, we don't know which one's male or female, so it doesn't matter which one we do on the top, although I do want the one with multiple different gametes to go across the top, because I like working from left to right. So folding system, exactly the same way as we did with dihybrid crosses. So this polygenic is done the same way to isolate the gametes, right? So if I have a capital C with capital B, that's going to be one of the gametes forming. Capital C, little b, different. And again, we don't show repeats within the same parent, and that is different, so we have to show that. Little c, little b, or capital B, sorry. Make my little c's very distinguishable from the big C's, so I don't mix them up. And the little c, little b, that's different. And of course, you remember what we said, that if a parent is heterozygous for both of those traits, that there are no shortcuts. You're going to have 4 different gametes forming. But with this one, you can see we're going to get some repeats. Little c, capital B, and everything else is a repeat. So we are going to simplify this and save some time on an exam by not showing repeated gametes. Okay, just like we did with other dihybrids or the previous question, now we bring the C's, the traits back together, the alleles back together. Bring the C's together, bring the B's together. So we have capital C. Always show the dominant allele first. Little c, capital B, capital B. The big C indicates that it is going to be colored. Pigment's going to be shown. And the big B tells us that this offspring is going to be black. Okay, and we just keep doing this. So now if this sperm is fertilized by this egg, we're going to have capital C, capital B, little b. That's also going to be black. And if the other sperm and egg come together in fertilization, little c, big B, big B. This one, as long as I have big C's, I don't care what the other ones are, this is going to be an albino. And same thing with this, little c, big B, little b. Because I have two little c's present, right, I'm going to be an albino. So my ratio is, out of the 4 blocks, 2 of them are going to be black, 2 of them are going to be albino. We must reduce ratios, reduce fractions, reduce everything to 1 and 1. And of course 1 being black and 1 being albino. Okay, any issues, give me an email and we can go over it. Thanks, guys.